Hello everyone. So last time in strain improvement we have been discussing about the three ways of modification. The first way of modification and its concerned example we have already studied, that is modification of permeability. Second way of modification under mutation and selection technique is isolation of auxotrophic mutants. You see that there are two types of organisms, prototropic and auxotrophic. When wild type or natural isolates, we call them as prototropic organisms. When they are mutated, they lack the ability of synthesis of particular compound. Such mutants are called as auxotrophic mutants. That means auxotropes are not able to synthesize particular type of growth factors. So they have to depend on external addition of these factors for their growth. In simple words, this modification is related to the isolation of mutants which do not produce feedback inhibitors or repressors. We have learned earlier that feedback inhibitors or repressors are responsible for limiting the production of microbial metabolites. Feedback control mechanisms are responsible for prevention of overproduction of microbial metabolites. That means these are the barriers for the overproduction. So, therefore, microbes are not able to produce these metabolites in excess. This is the way of modifying the organism such that the organism should overproduce the microbial metabolite. Now, particularly, we are talking about overproduction of primary metabolites. So, these are the mutants that do not produce inhibitors or repressors. What are these inhibitors or repressors? These are nothing but the microbial metabolites of the pathway, either it is unbranched pathway or branch pathway, and these metabolites are responsible for the feedback inhibition or repression. That means they are responsible for limiting their own production, controlling their own production, which is called as feedback. So in this type of modification, the control pathway is removed. That means if the organism is not producing inhibitor or repressor, then there is continuous production of microbial metabolites. Let us see how. This kind of modification, that is auxotrophic mutants, are useful for the production of intermediates of unbranched pathway. Production means overproduction. Second, auxotrophic mutants are useful for the overproduction of intermediates and end products of unbranched pathway. So you need to remember the difference between first sentence and second sentence. At the same time, if these inhibitors or repressors are essential for the growth, so as we are talking about primary metabolites, this means that if the organisms are not producing repressors or inhibitors, which are primary metabolites, so they are required for the growth. So if they are not producing, then they must be added to the medium in feed limiting concentration. Feed limiting concentration means the concentration which is only sufficient for the growth but not for the feedback control. It is called as feed limiting concentration. Now we have learned about the importance of blueprints in order to proceed for the strain improvement program. So blueprint, if the blueprint is available, that means if the biochemical pathway is understood, if the pathway is known, like whether it is unbranched pathway, whether it is branched pathway, and what are the feedback control mechanisms, which metabolites are involved in feedback control, which enzymes are involved in feedback control, the enzymes which are repressed or inhibited, such enzymes are called as key enzymes. So if the blueprint is available, then we can plan the changes for the possible modification of the organism, possible production of auxotrophic mutants for the overproduction of microbial metabolites. Now we'll see few hypothetical example based on the availability of these blueprints. First example is of unbranched pathway. So in this case, A, B, C, D and E are the microbial metabolites of this unbranched pathway. The dotted lines show feedback control mechanism. It may be inhibition or it may be repression. Here we can see that the enzyme required for conversion of A to B is indicated by dark circle, and this enzyme is sensitive to the levels of E. That means if the production of E is in excess, then this E will go and it will control the first enzyme, that will be called as key enzyme. So if this pathway is known and now we want to plan, we want to modify this organism for the overproduction of C. C is the intermediate of unbranched pathway. Then what would be our strategy? Our strategy would be that the organism should be modified such that it will lose its ability to synthesize the enzyme required for conversion of C to D. So that will be the possible site of mutation. Site of mutation means the enzyme responsible for the conversion of C to D, its set of genes is targeted, means mutation is created in the genes related to the production of enzymes responsible for conversion of C to D. That will be called as a site of possible mutation. So if that is the site of possible mutation, then there are chances of no production of feedback inhibitor or repressor, and if this pathway is blocked here, then it will result into overproduction of this intermediate called as C. At the same time, this organism will be called as an auxotrophic mutant. Why? Because now it is not able to synthesize E. E is a growth factor. So the organism will be auxotrophic for E. The care should be taken when this auxotrophic mutant is to be grown in the medium. It will require the addition of E in feed limiting concentration in the medium. That means its concentration should be such that it should support the growth of the organism and it should not be available for feedback inhibition of the first key enzyme. Second hypothetical example is of branched pathway. We can see here that A is getting converted to C through B. Then there is a branch point at C. C gets converted to D, subsequently to E, C gets converted to F and G. We see here that there is a concerted feedback control. For example, this end product of two branches E and G both have a combined action on the first enzyme that will be called as key enzyme required for conversion of A to B. Remember that it is a concerted, that means there is a combined action of E and G on the first enzyme. However, other feedback control systems are E is independently able to control the enzyme responsible for conversion of C to D. G is responsible for the controlling the enzyme responsible for conversion of C to F. Now, for example, after having this blueprint in front of us, now if we want the mutant is to be generated such that it should overproduce this C. So what should be the possible site of modification we can think of, we can plan, like if the organism loses or if it is mutated such that it will lose its ability to synthesize this enzyme responsible for conversion of C to D, then what will happen? There will be no more synthesis of E. In that case, C will accumulate. C gets further converted to F, F further gets converted to G. However, G is controlling its own production. Therefore, C will accumulate and there will be overproduction of C. In this case, this mutant will be auxotrophic for E because this mutant is no more synthesizing this E. So therefore, when we grow this organism in the medium, it requires the addition of E in feed limiting concentration in the medium. We can see its graphic like this. This is the pathway, and if this is the site of possible modification, then you can see the control mechanisms will be removed like this, and the mutant will have only this pathway from A to C, C to G. There will be control over production of G but no control over production of C. Third type of example is if we consider the same biochemical pathway and if we are interested. So in this case also E and G are responsible for concerted feedback control. There is a problem in this figure. It is showing independent control, but it is a combined action of E and G over the first key enzyme. If we think of site of modification at these red dots, and second site of modification is related to the synthesis of enzyme responsible for conversion of C to F. So this is the plan for overproduction of C. The possible mutant should overproduce this C metabolite, but in this case two sites are blocked. So this mutant will no more produce the end products of both the branches, and this is how the organism will be mutated like this. There will be conversion of A to B and B to C, and C will be overproduced. In this case, this mutant will be called as a double auxotrophic mutant, and it is not synthesizing two end products or two growth factors, that is E and G. Therefore, the care is to be taken that when the organism is grown in the medium, there should be addition of E and G in feed limiting concentration. Next example is, for example, considering the same branched biochemical pathway, if overproduction of F is intended and if the control systems are same, like E and G are responsible for concerted feedback control, E is responsible for independent control over enzyme responsible for conversion of C to D, and G is also have independent control. And if the overproduction of F is desired, then what is the possible way of generation of auxotrophic mutant? So the possible site of mutation should be the enzyme responsible for conversion of C to D and the enzyme responsible for conversion of F to G. In that case, there will be no production of feedback inhibitors or repressors like E and G, and the control over first enzyme will be removed like this, and this is how there will be overproduction of this F metabolite. In this case also, this mutant will be a double auxotrophic mutant that will require addition of E and G in the medium, of course in feed limiting concentration. The last example of this hypothetical pathway, if the pathway is more complex like this, in this case we can see that there is a branch point at C and another branch point at F, and if the control is like this, concerted feedback control by combined action of E and I. In this case, if the production of E is desirable, that means if we want overproduction of E, in that case this should be the site of modification. So that the whole pathway we will diverted towards the production of E, and there will be no more control on the first key enzyme. This auxotrophic mutant will require addition of G and I in feed limiting concentration. Therefore, the isolation of auxotrophic mutants may result in isolation of high producing strain, provided that the mutation for auxotrophy occurs at correct site. The recovery or isolation of auxotrophs is a very simple process. However, the difficult part is to recover the high producing auxotrophic mutants. Thus, the relevant auxotrophs may be isolated from the survivors of a mutation and subsequently screened for the productivity. This is an example of the method of isolation of auxotrophs. This method is called as penicillin selection technique for the isolation of auxotrophic mutant. In this case, the organisms or the wild types are exposed to mutagenic agent. So incubation with shaking conditions under desirable temperature for desirable period, and after that the cell suspension is exposed to the mutagenic agent, that is ultraviolet light. In this case, this protocol is standardized. Then after mutation, the organisms will die, and whatever the survivors are there, these survivors of mutation treatment are transferred into a complete medium. So this complete medium will support the growth of prototropes as well as auxotrophic mutants. After incubation, the cells are washed, and then from these cells it will be a mixture of cells, prototrophic cells as well as auxotrophic cells. Now the task is to separate auxotropes from prototrophic auxotrophic mutants from prototropes. For this, these cells are transferred into a minimal medium. Minimal medium means this is the medium that lacks the particular growth factor for which the auxotrophic mutants are auxotrophic. That means that particular medium component is missing. This means that minimal medium is not going to support the luxurious growth of auxotrophic mutant. Minimal medium will only support the growth of prototropes. During their transfer into minimal medium, this medium is treated with penicillin. Penicillin is an antibiotic which acts on growing cells, multiplying cells. Therefore, which cells are growing in this minimal medium? Prototropes. So prototropes will get killed due to the action of penicillin. After the incubation period, whatever the cells that remain alive are nothing but auxotrophic mutants. Then these auxotrophic mutants are transferred onto complete medium so that the auxotrophic mutants will now grow. This is how this process is repeated several times for the separation of auxotrophic mutants. Another technique for the isolation of auxotrophic mutants is replica plate technique. First, the survivors of mutation treatment are transferred to complete medium. That means on this medium, prototropes and auxotropes will grow. Then a replica of this plate is taken on the plate containing minimal medium. That means the organisms on complete medium are transferred onto minimal medium with the help of velvet pad. So on this medium only prototropes will grow. So the positions are already marked for the isolates on master plate, and these positions are then matched with the replica plate so that the organisms which are not growing on minimal medium but growing on complete medium will be the auxotrophic mutants. So after incubation, the colonies missing on minimal medium and present on complete medium are auxotrophic mutants. That is about the method of isolation of auxotrophic mutants. Now let us see an example of strain improvement, the use of auxotrophic mutants which are generated through this strain improvement program and which are useful for the overproduction of microbial metabolites. So this is an example of the use of auxotropes for the production of primary metabolites. Auxotrophic mutants of Corynebacterium glutamicum have been used for the production of lysine. So wild type Corynebacterium glutamicum is mutated to auxotrophic mutant of Corynebacterium glutamicum, which is the overproducer of lysine. The control of the production of this amino acid of aspartate family in Corynebacterium glutamicum is like this. That means this is the blueprint. It belongs to aspartate family. Aspartate is converted to aspartyl phosphate through aspartokinase. Aspartokinase is a key enzyme. Then this is converted to aspartyl semialdehyde. There is a branch point. Aspartyl semialdehyde gets converted to dihydrodepicolinate and homoserine. Dihydrodepicolinate then gets converted to lysine, and aspartyl semialdehyde gets converted to homoserine through homoserine dehydrogenase enzyme. This is another key enzyme that you need to remember. Homoserine further gets converted to homoserine phosphate, threonine, alpha ketoglutarate, and subsequently it gets converted to isoleucine. Homoserine, there is a branch point which leads to synthesis of one more amino acid, that is methionine. This is the biochemical pathway, and the feedback control mechanism is like this. Homoserine has an independent control over homoserine dehydrogenase enzyme. Threonine has independent control over homoserine dehydrogenase enzyme. Whereas one more control is existing here, that is threonine and lysine. They have a concerted feedback control over aspartokinase, like this. So threonine plus lysine, aspartokinase is sensitive to the combined action of threonine and lysine. Understanding this pathway, we can plan the possible modifications for the overproduction of lysine. What would be the possible modification? The possible site of modification should be at the branch point of aspartyl semialdehyde. That means this organism is modified such that it will not synthesize homoserine dehydrogenase. If homoserine dehydrogenase is not getting synthesized, then this mutant will be called as homoserine auxotroph. That means this organism will not be able to synthesize homoserine, and there will be no further formation of methionine, threonine, and isoleucine. So if that is the site of possible mutation, that is homoserine auxotrophs. So these homoserine auxotrophs will be the overproducers of lysine. The Japanese scientist Nakayama used this mutation and selection technique and selected a homoserine auxotroph of Corynebacterium glutamicum by penicillin selection and replica plate technique, which was an overproducer of lysine. This improved strain produced lysine in medium containing a low level feed limiting concentration of homoserine or threonine plus methionine. So if this mutant is a homoserine auxotroph, then there is need of addition of homoserine in feed limiting concentration, or there can be addition of threonine plus methionine in feed limiting concentration. The mutant lacked homoserine dehydrogenase, which is required for the synthesis of homoserine from aspartyl semialdehyde, thus no synthesis of threonine. In this way, concerted feedback inhibition of aspartokinase is removed. Therefore, aspartyl semialdehyde is converted solely to lysine.